Hello everyone, and welcome to our cumulative presentation for the 2021-2022 NASA Astrobiology Internship Program. I am Cameron Beeman, second year in the program, and I will be the Master of Ceremonies tonight. This program is a collaborative effort between Lassen Volcanic National Park, NASA's Ames Research Center, and Red Bluff High School to go beyond standard science classes and provide a unique learning experience. Tonight, we will be reviewing all that we have done in the program this year. To start us off, Olivia Palangoni will go over our outline for the night.
In this presentation, each intern will be explaining the key parts of our study and how they are interconnected. To begin, the concept of astrobiology will be explained and why Lassen Volcanic National Park is a part of our studies. Then the matter in which we collected samples will be explained. Next, each field site we studied will be described. Then the rock dissolution experiments we conducted in the classroom will also be outlined, along with the experimental parameters of our Winogrossi incubations. Lastly, the second-year interns will summarize the water chemistry at each site. Next, Hiram Bliss will be explaining what astrobiology is.
Astrobiology is the investigation of the origins, distribution, and future of life in the universe. A key aspect of astrobiology is to search for and characterize the full diversity of habitable environments, both on early Earth and on other planets. Volcanic activity and its interactions with water are key aspects of ancient habitable environments on Earth. Evidence of such interactions have been discovered on Mars, and these interactions probably also occurred on other Earth-like planets in the universe. And now Asael Diaz will be explaining why we are so interested specifically in the Lassen Volcanic National Park area.
Volcanic activity and its interactions with water were key components in allowing past life to live in their environment on Earth. From photographic evidence taken by our fellow robotic friends on Mars, we know that Lassen Volcanic National Park shares various geographical similarities with Mars, this being the aforementioned volcanic influence and effects of liquid erosion on the ground. The volcanoes in Lassen Volcanic National Park create unique opportunities for study. For example, groundwater in Lassen Volcanic National Park interacts with molten rock stemming from volcanoes. The subsequent groundwater, now altered by its previous contact, interacts with living organisms, microbes, and landscapes of its surrounding environment. Seeking to understand the mechanisms behind Lassen's wide range of environmental conditions and rich microbial life that are all impacted by the volcanic activity, we can replicate many of the hypothesized conditions of early Mars. The study of microbes in the present day can help answer the question, was there life on Mars? Up next, Todd will talk about the prerequisites for life to even exist.
In our studies of Lassen, we looked not only at the conditions of the water chemistry and geology, but also at the microbial life that is present. There are 4 categories that we observed to determine if the conditions are favorable for life to survive and prosper. The first of these 4 requirements is for life to have a solvent present. A solvent, typically that of water, is necessary in order to dissolve many nutrients for life. These essential elements for life which are dissolved by water include carbon, hydrogen, nitrogen, sulfur, and more. For life to be present in an ecosystem, a source of energy also must be present. Energy is provided to the organisms at these sites through the processes of photosynthesis, the absorption of light, and chemosynthesis, which provides energy through chemical reactions. The fourth and final necessity for life to exist is that of favorable conditions in terms of temperature, pH, and salinity. For life to exist, the conditions of the environment must be suited to what the microbes need. Many microbes have different adaptations to survive in different environments. For instance, many organisms can survive in an acidic environment, while others must be in a more neutral environment. On the whole, it is necessary for the 4 categories seen in the Venn diagram to be present for life to survive and grow. Now, Cameron will introduce the field sites that we studied at Lassen.
After learning about astrobiology and the requirements for life, we looked for field sites within Lassen Volcanic National Park to see if we could apply what we had learned. When observing Lassen, it is important to remember its history. Lassen Volcanic National Park once had an ancient volcano, named Mount Tehama. Over time, Mount Tehama eroded away from the inside because of the underground acidic hydrothermal activity, causing it to collapse in on itself. This volcano was massive, its footprint outlined in red. Within the remains of this ancient volcano, we see the Sulphur Works area circled in yellow. At Sulphurworks, we picked 3 significant field sites to observe: Ridge Lake Stream, Upper Pond, and Big Boiler. Now moving outside of the volcano, we see the Warner Valley area. Within Warner Valley, we picked 2 significant sites to observe: Paddlewell Creek and Alkaline Spring. We will now hear 5 of our first-year interns describe the field sites we observed in Lassen Volcanic National Park. First, we will hear from Mia Gleason.
Paddlewheel Creek is found in the Warner Valley area of Lassen Volcanic National Park. Paddlewheel Creek is a freshwater stream with conditions that can commonly be found at any other national park because they are moderate and relatively normal. When you approach Paddlewheel Creek, it appears to be a typical mountain stream that is thriving with an abundance of life. There is grass and trees to the edge of the stream, as can be seen in the photo, along with algae in the stream. The temperature and pH support this because the temperature of Paddlewheel Creek is 9 Celsius, which is an average temperature, and a pH of 6.7, which is neutral. The conductivity of Paddlewheel Creek is 52 microsiemens. There is no volcanic activity at the site because it is a freshwater stream. Up next, Natalie Moore will be introducing Ridge Lake Streams.
Ridge Lake Streams of Sulphurworks appears to be a typical mountain stream. It is fed by runoff from Ridge Lakes in Lassen Volcanic National Park. However, the water chemistry here tells a different story. The pH of 5.8 seems standard, but is low in comparison to places like Paddlewell Creek, making it slightly more acidic than that site. The conductivity of 290 is relatively high when compared as well. The stream is filled with lots of vegetation and algae. And now I'm introducing Courtney White to talk about Sulphurworks Upper Site.
Sulphurworks Upper Site is a hydrothermal feature at Lassen Volcanic National Park. Upper Site is a small acidic pool adjacent to Ridge Lake Stream with a low pH and a mild temperature. Upper Site has a temperature of 10 degrees Celsius or 50 degrees Fahrenheit and a pH value of 3.6. The pool appears to be bubbling and the reek of sulfur can be smelled when approaching the pool. As seen in the top right image, Upper Site has a beautiful vibrant top layer of green photosynthetic microbes. When removing the top layer of green microbes, a reddish-purple microbe layer can be observed below the green surface.
One iconic hydrothermal area found at Lassen is the Big Boiler found at Sulphurworks. When approaching the site, you'll notice the yellow-orange clay hillside surrounding the area and see little to no vegetation like plants or shrubs growing around. It also has a strong distinct smell of rotten eggs, which is sulfur, and has boiling mudclouds up close. Big Boiler has a high temperature of 107 degrees Fahrenheit found inside and an acidic pH concentration of 3, making the highest in all the field sites. This is because the groundwater reaches to a boil, causing the water to bubble and steam. The next site being talked about is Warner Valley Alkaline by Maya Rodriguez.
At first glance, the alkaline site in Warner Valley seemed to be a healthy stream, much like Paddlewheel Creek. However, looking closer, we saw it was much different. In these pictures, you could see a thermal image of the heat coming from the water, that there was steam coming off the water, little to no plant life, weathered rocks surrounding the stream, and thick green micromats, along with a few dead insects in the stream. Additionally, it has a temperature of 62 degrees Celsius, or 144 degrees Fahrenheit, a pH of 6.6, and a conductivity level of 1010. Together, this indicated a much different environment at the alkaline site than we have encountered in Lassen Volcanic National Park.
After viewing the field sites, we returned with questions regarding the various readings we took and observations we made. Why did we see the various differences in pH and temperature? And why did we observe differences in the life at each site? We set up several experiments to explain these differences. Now, Leah Moreno will give us a brief introduction to the lab environment and tools we used.
Using the meters shown, pH, conductivity, and temperature were measured from each site. pH is a measurement of acidity from 0 to 14. Sites at Lassen Volcanic National Park range between the acidic levels of 1.7 and 7. Conductivity is a measurement of the dissolved solids known as ions. Cations are positively charged and anions are negative. Sulfate is an anion we've measured from the sites produced when the volcanic gas hydrogen sulfide reacts with oxygen and water. This creates the acid that contributes to the acidity of sites and indicates the presence of volcanic activity.
We will now hear 2 of our first-year interns describe and discuss our findings from the water chemistry experiments we conducted in the lab. These experiments were designed to explain the interactions between rock and water of various pH and temperature values. First, we will hear from John Sheffield, who who will be discussing the effect rock has on the pH of a solution.
During our visitations at the Lassen Volcanic National Park field sites, we are presented with a question of how the pH at these different field sites will react with the rocks surrounding them. Our hypothesis for this question was that the pH of these field sites were dissolving the rock into the water, which raised the pH. To test our hypothesis, we set up 6 different testing stations that fluctuated in both pH and temperature. We then used the 2 rocks found in abundance near the Lassen Volcanic National Park field sites as rock powder. The data below shows that over our month-long experiment, we had to continually add sulfuric acid to the solution to lower the pH down to the desired pH. This can be seen in each line, with the green line needing 3,200 micromoles of sulfuric acid over 30 days to lower to the original pH, and the red line needing 9,700 micromoles. When observing the yellow and blue line, the pH of both is 5.5, which is closer to a neutral pH of 7. This results in their pH raising a much smaller amount each week when compared to the solutions with a pH of 2. Therefore, from these experiments, we can conclude that rock reacting with an acidic solution will raise the acidic solution's pH. I will now be introducing Aubrey Norton, who is presenting solubility of rock at different pH and temperatures.
We compared the change of net conductivity in experiments that have like pHs and different temperatures. We know that any difference in conductivity between the 2 experiments must be because of the change in temperature, because that is the only difference. The same goes for having like temperatures and different pHs. Net conductivity is the total amount of rock dissolved into solution during an experiment. We hypothesized that solutions with a lower pH were able to dissolve more rock into solution than that of a higher pH. We were able to test this through 2 experiments. One experiment had a pH of 2 and the other had a pH of 5, and both were at the same constant temperature. Our data shows that in fact the lower pH was able to dissolve more rock into solution. Our next hypothesis was that solutions with a higher temperature were able to dissolve more rock into solution than that of a lower temperature. We were able to test this through 2 experiments. One experiment had a temperature of 35 degrees Celsius, the other had a temperature of 20 degrees Celsius, and both were at the same constant pH. Our data shows that in fact the higher temperature was able to dissolve more rock into solution.
We will now hear 4 of our first-year interns describe and discuss our findings from the microbial incubations we conducted in the lab. These experiments were designed to explain and explore the effect of altering the various variables of a microbe's environment. First, we will hear from Sierra Lim on the effect of altering light.
We tested the impact of changing the color on microbes to see how much of the microbial life and diversity would be affected. To test this, microbes from the big boiler and alkyne sites were put into a container that only allowed blue light to transit through. This experiment gives us a better understanding on which microbes can survive in temperature-changing climates, which correlates to the past life on Mars. After examination, growth of the microbes was inhibited by the lack of light and energy that is typically provided primarily from red or orange color waves, which are being filtered out. However, some microbes were able to survive and be unaffected by the change, as they were more heavily rely on the blue light that was still present in the system, unlike some microbes that needed the red or orange light to prosper.
When we gathered the microbes from Lassen Volcanic National Park, we tested the microbes' ability to adapt to different environments. One of these environmental changes was the effects of temperature. We created 2 different Winograd columns to determine this effect. The first column was the control and the second was the experimental. The control was kept at room temperature, registering at 74 degrees Fahrenheit. The experiment was kept in an incubator registering at 105 degrees Fahrenheit. For the next 2 months, we observed any physical change to our columns. The first 2 pictures show the control of the experiment. The one on the left is the Winograd column, and the one on the right is the microbes under a microscope. You can see the biomass of the community is strong, and there are different types of microbes, which show that there's a high amount of biodiversity. The pictures on the bottom show the experimental column. Under the microscope, we can see that biodiversity is lacking. As you can see, the change is drastically different. The biomass of the community has greatly decreased, as well as the biodiversity. After the 2-month observation period, we concluded that when the temperature was either raised or lowered from the original temperature of the field sites, it would negatively affect the microbes both in biomass and in biodiversity. Now that we covered the effect of temperature on the microbes, Russ and Vaught will introduce the effects of pH.
After our visit to Lassen Volcanic National Park, we wanted to determine the extent of environmental changes these microbes can handle. We tested how pH would affect these microbes. Our initial hypothesis was that a change in pH in a microbe's environment would visually change their growth in abundance and diversity. During the experiment, we checked the pH of the control and experimental every Friday for the next 2 months. After experimenting, we determined that the original showed optimal growth in biomass and biodiversity, while the experimental showed little growth in biomass and biodiversity, resulting in us to believe that an increase or decrease in pH negatively affects the microbes in all aspects of life, as shown by the reduction of biodiversity and biomass. Now we will talk about the effects of change in concentration on the microbes.
Solute concentration, otherwise referred to as conductivity, is a measure of ions dissolved in solution. These ions are utilized by microbes as a source of nutrients or a fertilizer. Field sites in Lassen Volcanic National Park had a variety of ions dissolved in solution and a broad range of conductivity values. Certain microbes have specially evolved to survive in a specific set or threshold of conditions and have a threshold of solute concentrations that they can survive in. Our general hypothesis for lab experimentation was that a decrease in conductivity would lead to a decrease in microbial activity and vice versa, depending on the magnitude of the change. However, lab experimentation provided inconclusive results, as no apparent correlation between solute concentration and microbe activity could be found. Pictures of these lab cultures can be observed to the right, with the number of days, magnitude of change, and field site indicated.
Earlier, we had questions on why the various field sites had differences in pH, conductivity, and microbial life. After conducting our experiments and compiling our results, we now have answers to those questions. We have gained a new understanding of how Lassen Volcanic National Park works, and we feel confident that we can share that understanding with you. Now, you will hear all 5 of the second-year interns interpret the field sites with our new understanding.
As mentioned earlier, Padua Creek is a typical mountain stream that exists as snowmelt and rainfall flows through the valley. The pH of rainwater is around 5, while the pH of the stream is around 6.7. This change in pH is explained by our rock dissolution experiments, where we prove that the pH of a solution increases over time as rock is dissolved within it. We also saw in our rock dissolution experiments that environments with a neutral pH and cold temperature dissolve rock but at a fairly slow rate, which explains why the conductivity is so low and why there was not a large increase in pH from the original, as only a small amount of solutes were dissolved. Padua Creek also contains an extremely low level of sulfate, which is because of the lack of hydrothermal input seen at the site, once again confirming the simplicity of the creek. The microbes at Padua Creek, which primarily consist of a green algae, are able to survive and grow due to the mild conditions they live in. In the biology experiments previously discussed, these conditions of a neutral pH and cold temperature allow for steady growth, and a variety of diverse microbes are seen when these conditions are mimicked. Now, Asael will discuss the water chemistry found at the Ridge Lake stream of Sulphur Works.
The Ridge Lake stream resembles very much the image of Padua Creek, that of a typical mountain stream. Its main source is runoff from snowmelt and rainfall, and its low temperature of 40 degrees Fahrenheit is indicative of the cold climate typically seen. While the pH of Ridge Lake was measured at 5.8, we can assume that its actual pH would be closer to that of Padua Creek when accounting for the pH change from the snowstorm during our measurements. This is because, as stated before, our rock dissolution experiments indicated a rise in pH when rock dissolved within water, just like Padua. However, deviations start to appear when talking about conductivity and sulfate levels. Contrary to Padua's conductivity of only 52 microsiemens, Ridge Lake was at a low to moderate level of 290 microsiemens. Likewise, Ridge Lake had a relatively higher level of sulfate of 1,260 micromoles when compared to Padua Creek. As seen in our rock dissolution experiments, high levels of sulfate were suggestive of hydrothermal influence present within the Ridge Lake stream, indicating some sort of volcanic activity feeding into it. The presence of hydrothermal activity helps explain the estranged conductivity, where the solids from the source of the hydrothermal activity flowed into the stream to increase the conductivity to such a level. However, we are unsure if the sulfate comes from a vent directly connected to the stream or runoff from another site. Regarding the biology of Ridge Lake stream, there seems to be a relative amount of microbes subsisting well in their environment despite the higher than usual sulfate level. As seen in our microbe experiments, the moderate conductivity levels and neutral pH allows for steady and abundant growth. Up next is Cameron to discuss upper site.
The Sulphurworks Upper Pond is a field site with clear hydrothermal input. Here we saw bubbling in the pond, which one could assume means it had an elevated temperature. However, the pond had a cool temperature of 10 degrees Celsius, or 50 degrees Fahrenheit. This low temperature means that the bubbling is not a result of high temperature. We then can assume that this bubbling is a result of the release of volcanic gases. The pond had a relatively high sulfate level of 2010, and, as we already discussed, the high sulfate level is a result of volcanic gases, specifically hydrogen sulfide. This gas is a clear indication of hydrothermal input. The pond also had a low pH of 3.6. This is yet another indicator of hydrothermal activity, the low pH resulting from the present sulfuric acid. The upper pond also had a low to moderate conductivity of 385 microsiemens. This is consistent with our rock dissolution experiment, the low pH of 3.6 being more effective at dissolving rock. Despite the low pH, the upper pond is abundant in microbial life. One can visually see this by the distinct and vibrant green hue of the pond. However, these microbes are visually different from the microbes we saw at Paddlewell Creek, meaning the differences in water chemistry has influenced the microbial communities. Now, Hiram Bliss will describe to us the Sulphurworks Big Boiler site.
Seeing Big Boiler, it is a large pool of bubbling and hot mud. You can smell the distinct sulfur smell up close and easily see how volcanic the site is. As you can see, the mud pot has been practically carved into the hillside over time from the acidic environment and the hydrothermal activity. The reactions and conditions have altered the entire environment of that area, digging the hole of the mud pot, melting all the snow around the site, and creating that smell of sulfur. The sensory evidence, along with the sulfate level being so much higher than other sites, provide evidence of how hydrothermal activity is very active in Big Boiler. Despite the interactions Big Boiler has between rocks and water, like John spoke about earlier, as you can see, the pH is still pretty low due to the constant and active hydrothermal activity and the acidic environment that is keeping the pH as low as it is. Due to the low pH and the high temperature created by the hydrothermal activity, both of which work to increase the reaction rate and efficacy of the chemical reactions, Big Boiler has a really high conductivity, similar to what we would expect based on our rock dissolution experiments. High temperature and the low pH make solutions a lot better at reacting with the rocks. As evidence of this, at Big Boiler you can see the conductivity is so high, especially compared to the other sites like Paddlewell Creek that have more ambient temperatures and closer to neutral pH values. Another byproduct of Big Boiler being in this incredibly acidic and extreme environment and having a high temperature is the small amount of life around and in the mud pot. The microbes here consist of extremophiles, which are extremely tolerant microbes. This is because these microbes are the only life that are able to live in Big Boiler's intense environment. The high temperature and low pH make the conditions too harsh and inhospitable for most things to survive. Similar to how only a small amount of microbes can live in Big Boiler, we saw in our biology experiments that microbes in extremely high temperatures did not grow very well or died. Olivia will now explain the water chemistry and biological environment of our alkaline site.
As stated previously in the presentation, the alkaline site is a stream that runs through Warner Valley. Peering into the steaming water, one can observe that there are thick mats of green biomass. Water samples taken from the site indicate that the temperature of the water is 62 degrees Celsius or about 150 degrees Fahrenheit, and the amount of solutes dissolved in the spring is 1,010 microsiemens. The most shocking fact about the spring is that the pH recorded at the site is 6.6. This pH has about the same neutrality as bottled water. From our rock dissolution experiment, we hypothesized that solutions sitting at a high temperature would dissolve more solutes, which was proven to be correct. The high temperature at the site is the explaining factor into why the solute concentration is so high. The sulfate concentration of 1,690 micromoles and the high temperature indicate that there is hydrothermal output. A high conductivity and high sulfate levels is what would be typically recorded at a site with hydrothermal output, What makes the alkaline site so unique is that the pH is neutral, in contrast to sites like Big Boiler and Upper Pond. This can be explained in our rock dissolution experiment. As John stated earlier in our first hypothesis, as acid interacts with rocks, it raises the pH over long periods of time. For thousands of years, the water has been interacting with the rocks, raising the pH. This high pH is what allows the thick green mats of microbes to flourish, even under such high temperatures, which over time the microbes have evolved to be specifically adapted to withstand the heat. Lastly, Cameron Beeman will be explaining the significance of our studies.
Lassen Volcanic National Park is an example of how hydrothermal activity occurs on Earth and can sustain life in microbial communities. The discovery of ancient hydrothermal deposits on Mars indicates that some Martian environments might have sustained life in the past. The research at Lassen enhances our ability to search for and characterize any life that might have existed on ancient Mars. With that, we will conclude our presentation and move into a special portion of the night. To show our appreciation for the program and all involved, my fellow interns and I have prepared what we call impact statements. I will start us off. In case you missed it, my name is Cameron Beeman, and this has been my second year in the program. I have loved my time in the internship because of my love for science and learning, of which there was no short supply during my 2 years. Also, the opportunity to work with and learn from NASA scientists is phenomenal and completely unique to Red Bluff High School. I am grateful for the effort of the scientists, Lassen Volcanic National Park, and Mr. Michael for making this program possible.
Hi, my name is Todd Bell, and I have been an intern for this program over the last 2 years. And having taken this opportunity and working alongside these NASA scientists, conducting fieldwork, and learning whatever I could from them that I would have never experienced otherwise, has had a huge impact in preparing myself for my studies after high school and becoming a more well-rounded student for the future.
I feel a great amount of appreciation for how Mr. Michael and the NASA scientists have helped me to improve my knowledge of real experimentation and for how they've taught me to analyze and understand the environment around me.
My name is Olivia Palanconi, and I have been an intern in this program for the past 2 years. It has truly been a once-in-a-lifetime opportunity, and I'm so thankful for all that I've learned under the help of Mr. Michael and the NASA scientists. This program has really helped me solidify my passion for the STEM field, and I'm truly thankful for that. Thank you.
Hello, I am Marcelo Diaz, and thank you for watching our presentation that we all have worked hard for the last few years. It wouldn't have been possible without the grueling but very satisfactory education provided by Mr. DeMarais, Mrs. Parenteau, and Mr. Cuba. And thank you, Mr. Michael, for the supportive and very persevering attitude you had with us. I hope to get in my brain the mindset of a logical but empathetic person, like the way you guys showed us. Thank you.
Hi, my name is Ryan Wynn, and this is my first year in the NASA internship program. This program gave me valuable insight to what college-level studies entail by allowing me access to field sites that weren't open to the public to actually scoop for microbial life, which put me directly into the shoes of an actual scientist, which was pretty cool. It taught me many things that you really can't learn in your average science class, and studying next to real NASA scientists was an honor, and I'm truly grateful for that.
My name is Natalie Moore, and I'm a first-year intern, and the NASA program has provided me with a more hands-on experience and learning in a way that I've never experienced before. I'm very happy to be a part of it.
My name is John Sheffield. I'm a first-year intern in this NASA internship program. This program has taught me so much and allowed for a hands-on approach to science. For this, I thank all the NASA scientists and Mr. Michael for allowing such an amazing opportunity at Red Bluff High School.
My name is Leah Moreno, and I've been in the NASA internship program for the last year. This program has reminded me of the interests I had in space as a kid and given me the ability to understand the requirements for microbial life I hadn't known of before. It was an honor having the chance to be part of this, and I really appreciate all the time put into our learning by the NASA scientists and Mr. Michael.
Hi, my name is Sierra Lim, and I'm a first-year NASA intern. Throughout this program, I have learned so much about astrobiology and the importance of life. I was privileged enough to work with the most amazing scientists throughout this whole year. Through the in-person experiments and the live Zooms, I was able to get an experience I would have never got anywhere else.
Hi, my name is Courtney White and I'm a first-year NASA intern. This program is an amazing opportunity and has turned my love for science and space into an obsession. Since I was a kid, I have been very inquisitive about life, the universe, and how everything came to be. I want to know everything about everything. This program has allowed me to understand the biological, chemical, geological, and hydrothermal interactions that facilitate the conditions needed for life. I plan to study biochem in college to become an astrobiologist.
My name is Mia Gleason and I'm a first-year intern. Through this internship, I got the unique opportunity of learning more in depth about Lassen Volcanic National Park. I was also able to connect with what we were learning in class with what we saw happening in Lassen.
Hi, my name is Maya Rodriguez and I'm a first-year NASA intern. This program has exposed me to new things that I've never been exposed to in any of my other classes. Working with the NASA scientists has been a very enjoyable experience, as well as going to Lassen Volcanic National Park and seeing new sites that I've never seen before. Altogether, this is a one-in-a-lifetime opportunity, and I'm so glad that I got to experience this program.
My name is Mauricio Tamayo, and this is my first year of being in a NASA internship program. This program made me regain my knowledge of learning more about astrobiology. When I was younger, I remember being obsessed with space and learning more about basic biology in my elementary school. I always enjoy learning about science-related topics because I always learn something new along the way. And during this program, I learned that every little thing matters in our environment, no matter it's a big or small change. I hope later on in the future I get to learn more about our environment and our impacts.
Hi, my name is Justin Vaughn and I'm a first-year NASA intern. This program has taught me more than I could imagine. I had a fantastic experience collecting and growing microbes from Lassen Volcanic National Park. I also really enjoyed working with the NASA scientists and learning new things from them. And I would like to thank Mr. Michael for allowing me to be a part of this program.